40 carbon atoms long. It sticks off to the side. Idebenone has a chain that's only 10 carbon atoms long, and it has an alcohol in the end, which slightly changes its solubility characteristics. But it doesn't function as a CoQ10 replacement. So there was as a matter of fact, in preparing the next issue of the newsletter, there's a man who reported a case where he had angina, and he substituted idebenone for his CoQ10 and developed heart pains and had to go back to his CoQ10.
Yeah, that's the thing. Like, I myself, I'm very much of the belief that we ought to try to stay as close to the natural body chemistry as we can. And so when you get analogs, whether it's an analog to progesterone, which doesn't work, called Provera, or an analog to CoQ10, which who knows how that's going to work, why use it? We know what the natural stuff is. We can get it. So why not stick with it? Because after all, as Stan is always teaching us, balance is the name of the game. So if you get something and say, wow, this is 10 times as powerful as CoQ10, well, what's that going to do to the balance of the body? Something's got to be in balance with CoQ10, undoubtedly.
Maybe.
Maybe. So
I mean, there are significant advantages to the design of certain things like this that have been found. It doesn't, just because something is not natural doesn't mean it's not better. But there usually is some kind of consequence that if you don't look for it, you might miss it.
Sure. Well, I'm not saying that, that we should only use natural chemicals, natural or endogenous to the body. But until we know that some analog is really an improvement and safe, then why fool around with it? Just because someone says it's 10 times more powerful, that should be a red flag right there, in my opinion. I mean, you can even have too much antioxidants. A lot of important processes in the body are oxidative processes. And if you have, if you get super antioxidants that completely destroy free radicals in your body, you wouldn't even work right. Because that whole process is part of the natural way the body makes what it needs to make in order to keep in balance. So, yeah, Mike.
CoQ10, I've heard there's a new variety of that.
Excuse me?
There's a new version of CoQ10.
That's what he's talking about.
The gel, right?
Well, I don't know about that. Oh,
is that the gel?
A new delivery system?
Idebenone is not the gel.
No.
You're talking about a delivery system?
Yeah.
Yeah, I haven't heard that.
You heard about the gel?
No.
Someone said that it's a lot more effective at getting in the system.
Yeah.
So you don't take a lot less, cheaper. The other thing I wanted to announce is that followed Steve Richfield's protocol. Got my body temperature up.
Great. And what was it again?
Basically, he thinks he's figured out that there's kind of two temperature systems is his idea.
And better explain what's going on with thyroid.
Yeah,
this is cold and hot adaptation?
Yeah, probably. And it's to do with the thyroid, amongst other things. But the initial thing is that he discovered that his temperature was running low. He was running sort of about somewhere between 96 and 97 most of the time. And most of his family was in the same position. And so he was suffering under that. It was affecting him. And so he started doing research on it. And he got actually out onto the website a guy who did the thyroid.
Wilson?
Wilson, yeah, Wilson's disease. And ordered his manuals, the doctor's manual, and went through it. And Wilson had a program where you actually, you kind of used different T3, I think, and you ramp up T3. Right. But what he managed to do is he kind of adapted it a bit, and he thinks that what he did was he switched himself from being in a low temperature adaptive state to the 98.6 state by a careful protocol, which takes about a day to do. And so I followed his protocol. First of all, I had some normal thyroid, which is T3 and T4. ARMA, it's both. ARMA won't work. I took a whole bunch. I took so much ARMA that I should have been dead one day.
Armour thyroid, saying about armour thyroid.
Because I found I took some ARMA, my temperature would go up. I could get it up to 98, right? And for about half an hour, and I think it was the T3 that was working. And then it would start dropping back, so I'd take a bit more and go up to 98. And I took about a month's dose in one day, right?
And what was your heart rate doing?
Nothing.
Really?
I was perfectly fine. So apparently the reason for this is that there is a reverse T3 pathway, which in my system must be like this big.
Yeah.
And I just opened it up, right? And it just converted everything to reverse T3, which protected me. But it also meant that I couldn't get my temperature up. So when I then went off that and waited for a week or so and then just used just T3 in very small amounts of it, really, I was able to get my temperature going above 98. Right?
Amazing. It is interesting. So that's the kind of thing we want to keep track of. Anybody else? Something come up that they want to... I saw an article in the Mercury News today, which I put over here on diabetes, and they had a very large clinical study, and as you probably have seen for yourself by looking at that article, diet and exercise reduce the risk of diabetes and what do they call it, collateral damage, the consequential damage, by 58%. And this was not very extreme, as they point out, in terms of what people did on their diet, and they lost about, I think, 15 pounds on average, and their risk of serious diabetes type 2 was reduced by 58%. So I mean, diet and exercise, there's no substitute for it, really. Nothing that we can take is going to substitute for eating properly and getting regular exercise.
Eating properly is a big question mark.
Yeah, what eating properly means, of course, we all disagree about that one. And as a matter of fact, in that study, they used a low-fat diet, which, I think is not completely enlightened. The low-fat diet will work for many people, but I still think the zone diet is better. And but we won't get into that one tonight.
And does that apply to people who already have type 2?
Yes, absolutely, primarily. Yeah.
So that's what it's all about.
Yeah, that's what it's all about.
Much better results if you don't do the carbohydrate diet.
This
Much
thing
better
is at
results.
least, this shows, what this study really shows unambiguously is that what we're getting in the newspaper is somewhere between 10 and 20 years old. That is, the newspaper article today, there were studies that went back into the 80s and 90s. As I said when I gave that talk, the lady who had type 2, they were stabbing her every day with insulin. I looked it up. The literature at that time, we're talking about 1995, said that 6 out of 7 people could get rid of insulin and there's all signs of diabetes by diet and exercise. Now, she didn't. We really did it. That is, she got out and walked a mile a day or two, and lost 90 pounds, and lo and behold, all signs of it went away. But the point is that I then looked at the later literature about 1997. Along with the 1996 data, 1995 data said 6 out of 7, and around 1997, 98, they said about 9 out of 10. Now they come out with this thing, which is more of the same, but it's been going on for at least 10 years. And the reason for clubs like this is so that you don't have to wait for the Mercury News. We're hopefully getting the news 10 years ahead of time.
Right. Well, that's true. That's very true, Ira. And the only thing is, that we've only had, as long as I can remember, we've only had one fellow come and talk to us about exercise. That fellow from Stanford who came over about last year and did a really good job. And I just want to keep emphasizing that. I know that most people here are, believe in exercise, but I mean, we just can't underestimate that, because it isn't just a matter of the magic chemical and everything's going to be all right. So, but yeah.
It's probably not just simply a matter of exercise too. It's best to do exercise that you actually like to do.
Yeah, that's true.
Right.
And very few of us could enjoy the kind of hard work you do, Eric. How many miles you run this week?
By the way, just a little thing. There is a thing called the Senior Games in San Jose. I ran a race there. I ran a couple of races. I ran a 1,500 meter and a 400 meter. The 400 meter was really rough. I never run a 400 meter in my life. I usually ran, and it was a minute and 40 seconds, which was very slow. It's an embarrassment, but in any case, that was tough on me. And then I ran the 1,500 meter, and I got through in about 7 minutes and 25 seconds or so. And that at least both of them got me medals, largely because they were all, they couldn't get 70-year-olds.
You won medals in both?
Yeah,
Great.
I'm running a 5K this weekend. Again, part of that senior games business here. And I just got out and ran 3 miles before coming here, which is why I'm late.
So what did you, 70 what?
I'm 73.
73.
At this point.
Yeah.
And there are a whole list of things you get out of exercise, that some of which are very surprising. For example, gerontological community, in particular Dr. Bort, says a lot of the people in over 60 or so come in and say, you know, I'm tired. I'm tired. And what's the doctor supposed to do? He says, what he does, he gets them to exercise, and for some reason, from his viewpoint, it's a cure for tiredness.
Well, you know what?
And also in prevention today, CFS, which is chronic fatigue syndrome. A long article, or a page-long article, said that they found that exercise made a big difference
Mild
in
exercise.
mild exercise made a big difference in CFS. So there are all kinds of surprising effects of of exercise. But,
It's the principle, use it or lose it.
Yeah.
If you don't exercise, then your capacity for energy utilization and blood flow and all that diminishes.
Jim?
Yeah, I'd like to add here, Phil. I had a long talk on the phone one day with Dr. Ralph Holsworth, who's an MD, and he's now the consultant for the Essentia Water folks. The Essentia Water, where they electrolyze distilled or RO water and add minerals and then electrolyze it. And he had done a study that showed that if a person increases the pH of their overall system, that adult onset diabetes will often completely disappear. And that's what the Japanese have been doing for 40 years with these water ionizing devices. The theory being, and Steve might know more about this than I do, but that when the insulin receptor sites are in an acidic state, acid doesn't conduct electricity. Therefore, they don't send the signal to the brain, the insulin receptor sites, to have the pancreas release the proper amount of insulin. So rather than jam full of synthetic insulin, they found out that the symptoms of adult onset diabetes diminished or disappeared, and that the need for insulin dropped when the person got enough oxygen, and also which is one way of getting alkaline, and then just we're drinking the ionized alkaline water. Because when you walk, you take in 3 times more oxygen than when you're standing still, and then if you're outside, the number of molecules that have extra electrons on them means that oxygen is even more viable, so you have an antioxidant phenomenon.
Right. Okay, well that's very interesting. You know, well, I guess most of you know diabetes type 2 is I think pretty much identified with insulin resistance, and whether that's the best metaphor, I'm not sure, but at least the insulin doesn't work the way it's supposed to. You have high insulin in your blood, and it's not getting the job done of moving the sugar from your blood into the cells where energy can be produced. And you know, and the pH may be a factor. Exercise is a factor, maybe by introducing oxygen in part. There can be mineral deficiencies like chromium and vanadium and stuff that can be a factor in that and can be helpful. I wanted to say one thing about tiredness and exercise. You know, one of the reasons we get tired is because we don't get enough oxygen to our brain. And in fact, a lot of times if you're tired, you're just convinced you can't hardly stay awake. If you get up and breathe deeply, or even, you know, do some jumping jacks, you'll wake up like that in 30 seconds. And so exercise, your brain begins to get the oxygen it needs. Plus, when you exercise, your capillary system has to be responsive to the demands you're making on it, even mild exercise.
Have you ever tried vinegar?
You're talking about aerobic exercise, not...
I'm talking about aerobic exercise.
Bill?
Yeah.
Have you ever tried vinegar?
What about that?
It does the same thing to you. It's not necessarily, I mean, there's all kinds of complicated things in terms of saying, okay, acid, you know, produces problems at the cellular level, but I'll tell you, most people, if you take, you know, a sip of vinegar, it's like smoking a cigarette or drinking a cup of coffee or, yeah, I mean,
Of course, the vinegar is high in potassium. It's one of the highest foods, so that would increase your conductivity.
It's a very tricky business.
It has no potassium in it at all. It's just plain acetic acid.
Oh, really?
It's a fascinating
5%
subject.
acetic acid, no
Wow.
potassium, no nothing but acetic acid. It'll it's a very shallow acidifying agent to your bloodstream, but just acidifying your bloodstream has a focusing effect on the brain. It's pretty powerful.
Yeah.
You can do it with vinegar, and you can do it with an allergy. If you're allergic to something, just take a bite of something you're allergic to, and your brain sharpens up.
Wow. Oh, right.
Well, you know, Steve and I had a long discussion about 3 or 4 months ago because, you know, one of the interesting things that you probably have all have observed and sometimes been victimized by is that if you have a big lunch, all of a sudden you become, you know, like a half hour or 45 minutes after you've eaten a big lunch, you become overwhelmingly drowsy. I mean, I've had friends tell me that they're afraid they're going to fall asleep at a stoplight or things like that. And so what's going on there? So I did some research on that, and I had a long discussion with Steve. And it turns out that there's, you know, Steve has very strong and well-informed opinions, but I also saw some other things on the internet that I couldn't really figure it out. I mean, I kind of tend to believe what Steve comes up with, but there's still some unanswered questions because, well, I don't want to take any more time with it, but that's a fascinating issue. And if you look up on the internet, how did I search that? I searched. I don't know if you're interested. Call me, and I'll get my file out, and you can kind of check it out for yourself. But I mean,
the common opinion is that
Yeah.
the reason you get sleepy after you eat a big meal is because it's turkey, and turkey has tryptophan, and tryptophan puts you to sleep.
Right.
But that's just not
well,
right.
you know, the theory that I was working with was that when you eat a big meal, your body responds by secreting hydrochloric acid. So it draws acid from the rest of your body because hydrochloric acid is a very acidic acid and it's hard to produce. So the body, to be efficient, draws whatever acids it can that are available from the rest of the body, from the bloodstream and so forth. And when the bloodstream then becomes more basic, you get sleepy. Now, Steve says that's wrong.
No, I say that's right. That's exactly why you get sleepy.
Is that what you can see?
The alkaline tide of digestion.
Okay.
Your stomach gets acid, your body gets alkaline. That's how it stays balanced.
Okay,
It's
well, maybe that's
what
it.
do they call it? Conservation of pH.
Yeah,
You know?
but see, and that's why if you drink a little apple vinegar, what does that do? That introduces some acid into your stomach, makes it easier for your body to your stomach to become acidic, draws less acid out of your blood, and you don't get as sleepy. So, I don't know.
Why does the University of Maryland Medical School said if you take vitamin C prior to a heavy meal, a lot of vitamin C and vitamin E, vitamin E and vitamin C, that sleepiness does not take place, even though you eat a high-fat meal.
I'd like to get your reference on that.
Well, the University of Maryland.
I've taken big doses of vitamin C and I've never noticed that effect, but then I'd say that in terms of acidity, it would matter what kind of vitamin C it was. I mean, ascorbic acid is a pretty strong acid. So if you take 10 grams of ascorbic acid, that'll load up your stomach pretty strongly.
And do what?
My guess is ascorbic acid in that kind of dose would block the sleepiness effect. But if you did it as calcium ascorbate, it might not block it at all. It might make it worse. Or sodium ascorbate would make it worse, because those are alkaline forms of vitamin C, which would increase the demand for hydrochloric acid in your stomach.
I'd be curious to know what vitamin C Dr. Cathcart recommends as the vitamin C king of the year.
Well, I'll tell you what, why don't we wait till...
Always ascorbic acid, orally, or sodium ascorbate intravenous.
Okay, good, thank you.
You did bring this up. I always thought the reason you got sleepy after eating a big meal was reactive hypoglycemia, where the body makes a little bit too much insulin and that drops it. And I've had some success by starting people on chromium and or vanadium for that.
Yeah, I think that can certainly do it too.
Have you tried betaine hydrochloride with them?
Not for that, no. But
Yeah, it works pretty good. And any kind of acid load, vinegar during the meal will decrease that effect. So I think it's definitely a pH stress kind of response.
Yeah. Well, that's,
TMG, the
yeah.
same.
By the way, does everybody understand what reactive hypoglycemia is? You know, what happens is that you get a lot of food in your stomach, a lot of sugar in your blood. The body, in response, wants to move that sugar into the cells, so it secretes a lot of insulin. And it just secretes a lot. Sometimes when it's doing that big a job, it secretes a little bit too much. And then the sugar is brought out of the bloodstream, and there's an overswing. It takes too much sugar out of the bloodstream. As a result, you're now in a low sugar state. And what happens when you have low sugar? You get tired.
Your brain shuts down.
But I'll tell you one thing. That's interesting about that, Bob, is that when I get drowsy in the afternoon, I usually don't get crabby. But when I get hypoglycemic, I do get crabby. So I'm not sure it's the same for us.
Now, wait a minute. See, the body then counters the hypoglycemia by putting out adrenaline or cortisone to release the glycogen in the liver, and the adrenaline makes you crabby.
Yeah, I know, but the thing is, what I'm saying is, when I get this after lunch drowsiness from a big meal, I don't get crabby
at all, ever.
Right.
It's different.
So that makes me think it might be a little different. I don't know. But I'm sure, though, you're right, that the tiredness can definitely be brought on by response. Well, now let me just announce a couple of things, and then we'll get into the program because we want to leave time for the videotapes too. And I want to say a little bit about vitamin D next week. Now, you know, I'm a tremendous believer in nutritional medicine. I mean, I've been hanging out with you folks for all these years, and I've been reading on this stuff, and I'm amazed by the potency of vitamins. And of course, vitamins aren't very sexy. So I want to say a little bit about vitamin D and why you should all be here next month. You know, for a long time, vitamin K was thought to be a, very uninteresting vitamin. And then now they found out that it has to do with the body's use and allocation of calcium to various functions, and that it's really important. Now, something similar is happening with vitamin D, because there's a whole lot of new research coming out that says that most people in a civilized society have a vitamin D deficiency, and all kinds of things can be traced back to that. In the last, I guess it was New Scientist comes out, I guess, every week. I guess it was 2 weeks ago. New Scientist, which is this tremendous magazine out of Great Britain. It costs a fortune. It's like $149 a year to subscribe to it, but I guarantee that if you subscribe to that magazine, you'll read every issue from cover to cover. It is such a fascinating magazine. In that magazine, they had an article on schizophrenia and vitamin D. What's the connection? Turns out that schizophrenic persons, people who grow up to become schizophrenic, I think usually I'm not sure about this, I think the onset of schizophrenia is usually in late teens or something like that. But if you look back, try to understand what's going on in those persons' lives, you find out that 20% more of those people were born in March and in the months just on each side of March than you would expect to see born in that month. So why is it that people who are born in March have this high risk of schizophrenia? Well, they thought, well, maybe it has something to do with vitamin D, because as we know, if your mother is pregnant in the winter months, she's likely to not be getting very much vitamin D, because of course there's not that much sunshine, and we get vitamin D when ultraviolet B, which is a kind of elusive wavelength, which is not found except in the middle of the day with direct sun, because it doesn't penetrate the atmosphere very well. If it comes in at an angle, it usually gets absorbed by the atmosphere. If it's a smoggy day, you don't get it. It has to be a clear day and the sun pretty much right above before you get UVB. So in the winter months, you don't get much. And of course, most people in civilized society don't go outside. So what happens? The mothers have a vitamin D deficiency. So they started trying to check this out and look for other things, and they found out that the schizophrenic population can also be correlated with the latitude of the birth. The further north you go, the more schizophrenics are born, especially in the northern hemisphere. In the southern hemisphere is a little different situation because the light is a little more direct for some reason in the southern hemisphere than it is in the northern hemisphere. But in the northern hemisphere, it correlates with latitude. So then they started looking at mechanisms, and they come up with some mechanisms why this would be so. And so it looks as if a major factor in schizophrenia is going to be vitamin D deficiency, or in other words, a deficiency of sunlight. And so Clara Felix is going to be our speaker, and she's gotten onto this. And she's in touch with this woman, Kristen Sullivan, who's doing this tremendous synoptic research, putting together all the research on vitamin D, and she's writing a book about it. I invited her to come and speak to us, but she's so engrossed in getting this book out. She said, when I get the book out, then I'll come and talk to you. So we probably will hear from her. But you know, the interesting thing about vitamin D is that it relates to the immune system, it relates to cancer. D3 and cancer are, inversely related. And here's something very striking. I heard someone told me this. I think maybe it was Dan Field. I don't know who it was. Told me. I pretty much think it was Dan. He said, Phil, there's a correlation between the incidence of prostate cancer and latitude. Stan told me that. Is that right, Stan? Wasn't that you told me that? That's right. And I thought I could hardly believe it. This is before I read anything about schizophrenia and latitude. Yeah.
Latitude at time of birth or latitude at where you live?
No, this is latitude where you live now for prostate cancer.
That's the effect.
Sure. With schizophrenia, it's birth, but with prostate cancer, it's latitude. And I couldn't believe it. So I went on the web and I put in there prostate cancer and latitude. Now, who would ever think that something would come up, an article that would mention both? Wow, there's a whole big research story out there on latitude and prostate cancer. And it says, and the results are striking. I mean, study after study has showed that this correlation exists.
What's the correlation?
Between the further north you go, the more likely you are to get prostate cancer. What else correlates with prostate cancer, and therefore with vitamin D? Pigmentation. Black people get 3 or 4 times as much prostate cancer as we do, as white people. Why? Because their skin is designed to have a pigment which protects them from getting too much UVB, which you get if you live close to the equator. And then, of course, as you probably know, I hope you do, the reason we're white is because our ancestors migrated to the northern climes, and only our ancestors who were of that, kind of minority that were light-skinned survived. Our dark-skinned ancestors who went to northern climes died of rickets and probably prostate cancer and every other thing that you get if you don't have vitamin D. So I won't say much more about it, but any more about it. But but come and hear about vitamin D. This is exciting stuff. Okay. Now
I'd like to add that
yeah.
I take bone up, and bone up is just to prevent osteoporosis, and one of the ingredients is vitamin D.
Mm-hmm.
And vitamin D is involved in building bones.
Mm-hmm. Sure. Well, that's been on for a long time that vitamin D was involved in building bones. It's very important, absolutely important. And I don't want to minimize that. But it's also, it turns out, important in brain development. It's important in the function of the immune system. And so, and you see, the thing about vitamin D that's so exciting is, hey, we have to get outside. I mean, like, Claire keeps saying she just thinks she's going to have to become a nudist. Because,
only at noon, though.
Nudists at noon. Then we have a new society. Nudists at noon,
And how old is she?
Clara is in her 70s.
79.
79, that's right, 79, yeah. And you will not find a more vital or more brilliant 79-year-old person, I think. So if you haven't heard her speak, we shouldn't be here. Okay, now I want to bring on the main event here.
You'll be sunning, right?
Instead of mooning.
Instead of mooning, you'll be sunning.
I want to say that, too much sunlight causes wrinkles, and the ladies might worry about that. And I want to put a plug in for cod liver oil.
Absolutely.
Yeah. If you get your vitamin D through a source other than sunlight, your skin will look a lot better.
Yeah, that's true. And Clara's other big love is omega-3. Cod liver oil is great source of omega-3. So I mean, I've been taking my cod liver oil faithfully, I can tell you that. Dr. Alexander, if you don't like the taste of it, as Steve taught me, it only tastes bad if it's rancid. If you get a good preparation, it doesn't taste great, but it doesn't taste bad either. And if you get Dr. Alexander's, you can get it with peppermint, you can get it with this or that. And I've got it every which way, and I like that. I mean, I take the
That's the Norwegian.
big tablespoon and a half every day.
That's Norwegian, Alexander's
Yeah.
Norwegian.
Yeah. Stanley?
I talked to Clara about that, and she said even if you take cod liver oil, you still need the sun to activate it.
Well, this we'll find out. You know, we'll also find out about, you know, the cancer risks and stuff, which are very much overplayed. Everyone's afraid to go out in the sun now, which is just the terrible, terrible thing. It's like the big cholesterol scare. Everybody ran the wrong direction, sort of, you know. And now we're running away from sunlight, and we all need it. So, okay. Now, in order to give Steve a proper introduction, I want to introduce you to a man who's going to introduce Steve. This is Tim Gallagher, who many of you already know. Tim is one of our board members, and although he doesn't speak very often, I've discovered he's one of our most knowledgeable board members, and I'm very impressed with every person on our board, so you can imagine. Tim is a doctor, a dentist who specializes in the removal of mercury amalgams, so he knows a whole lot about this subject we're talking about. And he's brought a couple of videotapes which he can tell you about it and bring into the program at some point. I hope you figured out when, because you guys have to coordinate that. And on top of that, he's Steve's dentist.
We're still awake.
So,
lights.
So, Tim.
Yeah. Okay, tonight's lecture is Mercury Toxicity and Alzheimer's Disease with Steve Fowkes, and I'm sure all of you know him. He's past president of Smart Life Forum and editor of SERI. And that's the Smart Life News is the newsletter that he edits every month.
Everybody have a copy?
Yeah. How often do you come out with that?
It's irregular. SERI's about to go through some major changes right now, and so anybody who's a subscriber will hear about it.
Yeah, but he does a great job with his research and spelling it out thoroughly. And if you compare it to Ray Pete's newsletter, I mean, you read his newsletter and you're just totally confused, and you're so outstanding what you do. His talk tonight is based on studies that were done by Boyd Haley and the University of Calgary. And the first video is from the University of Calgary on neurofibril bundles, what happens to them when they're exposed to mercury. And they want to start out with that first because it's short, 10 minutes, and it kind of explains it in cartoon fashion, and it's very simple.
Mm-hmm. I think it's a good idea. Where's the popcorn?
With the butter.
How mercury causes brain neuron degeneration. Mercury has long been known to be a potent neurotoxic substance, whether it is inhaled or consumed in the diet as a food contaminant. Over the past 15 years, medical research laboratories have established that dental amalgam tooth fillings are a major contributor to mercury body burden. In 1997, a team of research scientists demonstrated that mercury vapor inhalation by animals produced a molecular lesion in brain protein metabolism, which was similar to a lesion seen in 80% of Alzheimer's diseased brains. Recently completed experiments by scientists at the University of Calgary's Faculty of Medicine now reveal, with direct visual evidence from brain neuron tissue cultures, how mercury ions actually alter the cell membrane structure of developing neurons. To better understand mercury's effect on the brain, let us first illustrate what brain neurons look like and how they grow. In this animation, we see 3 brain neurons growing in a tissue culture, each with a central cell body and numerous neural processes. At the end of each neurite is a growth cone where structural proteins are assembled to form the cell membrane. 2 principal proteins involved in growth cone function are actin, which is responsible for the pulsating motion seen here, and tubulin, a major structural component of the neurite membrane. During normal cell growth, tubulin molecules link together end to end to form microtubules, which surround neurofibrils, another structural protein component of the neuronal axon. Shown here is the neurite of a live neuron isolated from snail brain tissue, displaying linear growth due to growth cone activity. It is important to note that growth cones in all animal species, ranging from snails to humans, have identical structural and behavioral characteristics and use proteins of virtually identical composition. In this experiment, neurons also isolated from snail brain tissue were grown in culture for several days, after which very low concentrations of mercury were added to the culture medium for 20 minutes. Over the next 30 minutes, the neurite membrane underwent rapid degeneration, leaving behind the denuded neurofibrils seen here. In contrast, other heavy metals added at this same concentration, such as aluminum, lead, cadmium, and manganese, did not produce this effect. To understand how mercury causes this degeneration, let us return to our illustration. As mentioned before, tubulin proteins link together during normal cell growth to form the microtubules which support the neurite structure. When mercury ions are introduced into the culture medium, they infiltrate the cell and bind themselves to newly synthesized tubulin molecules. More specifically, the mercury ions attach themselves to the binding site reserved for guanosine triphosphate, or GTP, on the beta subunit of the affected tubulin molecules. Since bound GTP normally provides the energy which allows tubulin molecules to attach to one another, mercury ions bound to these sites prevent tubulin proteins from linking together. Consequently, the neurite's microtubules begin to disassemble into free tubulin molecules, leaving the neurite stripped of its supporting structure. Ultimately, both the developing neurite and its growth cone collapse, and some denuded neurofibrils form aggregates or tangles, as depicted here. Shown here is a neurite growth cone stained specifically for tubulin and actin, before and after mercury exposure. Note that the mercury has caused disintegration of tubulin microtubule structure. These new findings reveal important visual evidence as to how mercury causes neurodegeneration. More importantly, the study provides the first direct evidence that low-level mercury exposure is indeed a precipitating factor that can initiate this neurodegenerative process within the brain.
That's a bombshell. It's going to change politics around the world in a big way. But you might not see this on Discovery Channel or on CNN for 5 or 10 years.
Why?
Because mercury is a politically protected subject.
Is a copy of that film available?
Yeah, there's a website on the end of the tape that gave it.
You're giving a new meaning to political correctness.
Yeah.
Do you want the overhead set up there?
Yes. I mean, I think it's probably, if we can get an image over here, that's probably a good location.
Okay, well maybe we could put the pass this around and it tells you where you can get the video. Let me see.
I only caught part of it.
You got quiet there?
You know what? This is put
Maybe
together.
something.
Common,
Let's see if
UC
we
Calgary.
can-
That's about as much
we
as
have
I
to do it
got.
in the middle.
Steve, have you heard of that book, The Energy of Life?
You know, these symptoms that people show, and how are these things related to each other? And there has never been a comprehensive model that ties all these pieces together until now. And in terms of the politics of why this has happened, it's because there are people out there who are not following, you know, what I would say politically correct lines of research. This is not funded by NIH grants. This is not the kind of stuff that the drug companies are doing. As a matter of fact, I think there's somebody's in one article said that about $500 million is being spent by various drug companies trying to figure out how to block beta amyloid production, which is one of the proteins that accumulates in Alzheimer's disease, with the idea that if you can block this, this would be a viable therapy for Alzheimer's disease. Well, when you look at all these mechanisms that we've discovered here and you watch what mercury does to the systems, you find out that, you know, first glutathione is damaged, and then sulfhydryl enzymes are damaged, and then these microtubules disintegrate, and then beta amyloid gets produced. So your system has fallen apart before beta amyloid ever gets involved, and so stopping beta amyloid is like too late.
cheating this.
Cow's already out of the barn.
That's right. Yeah, everything is falling apart. And I mean, before everybody gets really down on mercury and starts thinking, you know, that this is all a major conspiracy to kill everybody, I would point out that this is not, Alzheimer's disease is not really an issue of mercury exposure. This is not the kind of thing that if you avoid mercury, you're not going to get Alzheimer's disease. That's not how it works. I'd say 99% of the people in the world today already have enough mercury in their brains to cause Alzheimer's disease. As a matter of fact, most babies are born with enough mercury to cause Alzheimer's disease. So what is it about mercury that poses this problem? Is because the mercury in some people is tied up, and in other people it's not tied up. And so the mercury gets loose and poisons certain enzyme systems. The brain develops Alzheimer's disease. In other people where the mercury is properly tied up, the mercury is not toxic and the brain is able to function quite well. Levels of mercury can be, in terms of words that are used by scientists to describe it, we call, the scientists in this particular study were using what are called nanomolar levels of mercury. And nano means 10 to the minus 9, which is 1,000th of 1,000th of 1,000th. Okay? But there are people who have diseases where scientists have measured millimolar levels of mercury. There's a kind of heart myopathy where they've done biopsies in living human beings and found levels of mercury almost, well, above millimolar levels, or at about millimolar levels, which is 1,000 to 10,000 times greater than what's producing Alzheimer's disease-like reactions in these neurons. So there is an immense capacity for detoxifying mercury in the human body. But if that system fails, you're in trouble. The bad news about my talk tonight is that I can't tell you how to cure Alzheimer's disease. I can't tell you what the best therapy is for resolving this kind of this mercury toxicity problem, because it has more to do with the biological defense mechanisms for mercury than it does with the presence of mercury itself.
Steve, is that why it's related to old age, because your defense systems are going down there?
Yeah, I think it's possible that Alzheimer's disease happens in children. We just call it autism. Autism. So, and the difference is that when people get older, their systems, their mercury detoxification systems are at jeopardy. And in children, they're not in jeopardy, but we give the kids doses of mercury in vaccines. And so that causes it overloads their systems and they develop this dysfunctional problem. And it may just be pure mercury toxicity, and maybe a combination of effects that combine to produce the autistic reactions.
In my talk last month, that was a result of vaccination of children was autism.
How's that?
And because the vaccines are preserved with mercury.
By the what?
Yeah.
Yeah. Do you know where my transparency is?
Yeah.
Is that the vaccine?
No, they were sitting out here on this table, and now they're gone.
Did they get carried away with the other machine?
Yeah.
No. Peanuts in here.
Did they scoot over here somewhere?
I have not seen.
Oh, wait, maybe I got it.
Peanuts?
You know what?
It's kind of hard to remember.
He's got them. Okay, good.
That's my strategy.
Okay, do you remember that illustration on there about microtubules? Okay, this is very nice, yeah. Cool. If this is not going to work, it's going to slide or something. How about,
where's the focus thing?
how about
You roast that little knob on the side there goes up and down, not up there, up on that shaft.
Oh, right, there you go.
Then you could tilt the mirror.
Okay.
Okay, filter thing.
Good, you got it. There you go. Excellent.
Great.
Okay, this is a microtubule here, and I have the alpha tubulin and beta tubulin subunits in different colors, so you can see that it kind of forms this spiral. And if you look at the cross-section, it's hollow inside. Each one of these little protein molecules links up with GTP, guanosine triphosphate, which is a chemical cousin of ATP. And ATP is the primary energy currency of the cell, and GTP is like this special currency that's used to manage the cytoskeleton, or the structural components within the cell that determine its three-dimensional shape. And microtubules are not only involved in shape, there are other fibers that are involved in maintaining the shape of the neuron. They mentioned, was it actin? But there are several different fibers. But the microtubules turn out to be very critical in several major respects in terms of Alzheimer's disease. Microtubules are, for one thing, they're just straight fibers, or like straws. They're completely linear. They don't branch. So when your brain is developing in utero, you have neurons in different parts of the brain that send out these long projections to go, and the instruction is grow in a straight line until you run into something. So if it's going past a dopaminergic neuron, it keeps going. If it's going past a serotonergic neuron, it keeps going. When it hits a cholinergic neuron, it goes, okay, I'm here. And then it stops growing, and it forms a nerve terminal. Now you've got a connection between this part of the brain and the cholinergic neurons in that part of the brain. And the microtubules are what make that happen, because they just grow in a straight line. And they'll grow as long as you want. Now, when you get to a point where the neuron meets something that it wants, it doesn't have to necessarily stop growing. It can just form a node at that point, and then starts several new microtubules that start growing from there in several different directions, and then it branches, and it branches, and it branches. And then, after you're born, your nerves are then pruned to make your mind from a random connection of interconnected, neurons to something that actually functions well. So if a neuron connects to something that doesn't provide a signal that relates to its needs, that gets pruned off and it dies and it disappears. And other sections can grow out and attach to things. And neurons that form links to something that does mean something, it does connect, you can have those branch and branch again and develop better and better connections. And a huge amount of neurons die between birth and age 2 years of age. But that makes us intelligent at the same time. So it's...
What happens to the neuron die-off? Is that just absorbed into the body?
Yeah. All the raw materials, it's all disassembled and is used to feed other neurons. And there are neurons that are growing in the process. So, and neurons parts of neurons may disappear, and the neuron stays alive. It's just, if it's connected up to something that's a dud, it disintegrates, and if it's connected up to something that's important, it's then reinforced. And the nervous system is built to operate on this way. And so that's probably why brains develop better and you get more intelligence if they're in a stimulating environment, is because all of these feedback connections that are saying what's a good connection, what's a bad connection, it's optimally being defined to those cells. Now, each one of these linkages here between beta tubulin and alpha tubulin requires a GTP molecule. So it's one unit of energy assembles them together. And up here we have, the random parts, and then some of the GTP is added and they start linking up. Here you have a white and a blue unit linking up to form these pairs. And then down here, the pairs link up, also using GTP, to form these protofilaments. And then down here, the protofilaments bind together, and there are 13 of them in a ring around it, and that forms the tubule. And it makes it very, very rigid, so it has very, very long-term structural stability. So what happens when you're exposed to mercury? Not only can the microtubules not grow, which is one aspect that happens at the lowest level of mercury exposure, but the mercury poisons the bonds that are formed here, because these things assemble and reassemble and disassemble fairly regularly and spontaneously. The large-scale structure-determining components in the brain disappear. So you've got this neuron with all these very, very long arms reaching all over the place. And all of a sudden, the structure goes away. That means these neurons can break, they can decompose, they can degenerate, and all of these interconnected functions in the brain become at risk. And that's why we have memory problems and other kinds of difficulties with Alzheimer's disease. Although I'll spend most of my time talking about Alzheimer's disease and mercury for this talk, this is possibly applicable to all neurodegenerative diseases. So if the cerebral cortex is affected, we call it Alzheimer's disease, and you get memory problems. If the cerebellum is affected, you get Parkinson's disease, or some other kind of dyskinesia, where motor control functions are impaired. And you can have spinal cord problems, and that may result in MS or some other kind of symptom like that. So many different neurological conditions may fundamentally relate to the destabilizing effect of mercury on microtubules. Any questions about that? Yes.
And so what you're saying is if the mercury is free or loose in the system, it may have a tendency to go to different areas which cause these different symptoms.
Right. If you have like mercury and mercury sponges, and they're equal, then the mercury is, most of it's in the sponges. But if you lose track of your sponges, and in one part of your brain you have fewer sponges in some other part of the brain, that means you have more mercury, loose mercury in that part of your brain.
I'm going to go there with the talking about how that might occur, whenever you have a more susceptibility or...
The basic antagonism that we're talking about here with mercury has to do with sulfur. Mercury and sulfur bind very strongly to each other. If you were to go out and study mercury mining in the world, you'll find that it's mercury sulfide, which is the primary ore that's found in the Earth's crust. And mercury also binds to sulfhydryl compounds in the human body. Very, very strong interaction there. And compounds that contain sulfhydryl bonds would be the most basic one, would probably be glutathione. How many people have heard of glutathione? Yeah. It's a primary cellular antioxidant in the water-soluble part of the cell. It works with vitamin C. So glutathione and vitamin C as a partner defend against the most basic kind of stress that we have in the world today, which is oxygen-related toxicity. Free radicals, oxygen radicals. And so if you have an oxygen radical in your body, chances are it's going to react with a glutathione molecule more than anything else. It's a big target, and it's specifically there for that purpose. And so we have fairly high levels of glutathione. So the relative toxicity of mercury is probably related to the ratio between mercury and glutathione. Now, when they studied the movies of these enzyme effects and these neurofibrillary tangles that form with mercury, the first thing that happens is that glutathione becomes depleted. So you expose the cell to mercury, and glutathione levels crash very, very quickly. And then, after a while, various enzyme systems start to malfunction. It's probably because the primary defense system is glutathione, and when glutathione drops, it's no longer able to bind the mercury, and the mercury is then slipping past the defense and sabotaging the microtubules. Okay?
So it's a stepping stone, sort of, or it's a pathway to that.
And glutathione synthesis is a basic regulated process, and we make glutathione out of cysteine and glutamine and lysine. And so if you have a shortage of any of those amino acids, you can have a problem. Or if your regulating system for synthesizing it gets the wrong feedback signals, you could have a problem. And there may be natural variation in the production based on your genetics, diet, environment. Yes?
Steve, I was reading that glutathione does not cross the blood-brain barrier because it's too big a molecule. So that means that the 3 amino acids have to be assembled inside the brain somewhere.
Right. That's a cellular process. Protein synthesis, glutathione synthesis is a cellular process. So each cell is manufacturing glutathione based on its own idea of what the proper amount is.
Wow.
Right?
And that could be where the genetic comes in to give it the command to be able to know how much to make or not to make.
Or what part of your brain fails first if exposed to mercury toxicity.
It's part of the genetics that affect that.
Yeah. Yeah.
Can you take glutathione to?
Yeah, glutathione, you can eat it. It's a dietary supplement sold in the United States over the counter. You can also consume the amino acids from which it's made. And if you eat, protein in food, you're eating those amino acids from which glutathione is made. But the rarest one is probably cysteine, which is the actual sulfhydryl compound that it contains the SH bond that would interact with the mercury.
One more thing on that one, Steve. In Blaylock's book on excitotoxin, he says that
cysteine is
L-cysteine is excitotoxin, but normal acetylcysteine is not.
I don't believe that.
Okay,
Yeah.
that's.
Yeah.
You think they're both excitotoxin?
Well, I don't know the pathway of that excitotoxicity, so I can't say that. But I do know that the SH group is not blocked in N-acetylcysteine. It's perfectly functional in N-acetylcysteine as it is in cysteine and in glutathione.
Yeah.
All three of them bind mercury and detoxify mercury equally well. So it doesn't have to be in the glutathione form to have antioxidant properties. But the problem is that because cysteine binds to mercury, if you put cysteine and mercury together, you now have a bound structure, and the cysteine will carry mercury wherever it goes. And so does this benefit the brain by putting cysteine into the body if it carries mercury into the brain, or does it go into the brain and then help carry mercury out? It could go either way. And since we don't have good ways of monitoring mercury exposure in the brain, one has to be careful about trying to control mercury without knowing that this is going to be effective, because you could theoretically produce the exact opposite effect that you wanted. Yes?
Because of this, I have some experience now with autism, and there's this lady, Dr. Amy Holmes, who is doing a tremendous amount of work with this. And it's sort of the ritual now that you do a urine mercury, and then you give them DMSA, which is
oral?
Orally, yes. And you keep that up until, and there's a formula to tell you the amount, and you keep that up until their urine mercury is down to about 1/10 of what it was before. And then you give alpha lipoic acid. Apparently alpha lipoic acid passes the blood-brain barrier and takes the mercury out of there. Now the reason why we do use the DMSA first is we want to get it out of the body mostly, because the DMSA could, or the alpha lipoic acid crossing the blood-brain barrier could take it into the brain. So there's a whole ritual involved in that.
Yeah, but
And alpha lipoic acid is also, in its oxidized state, it doesn't bind mercury very well at all, but in its reduced state is a very powerful mercury binder.
Of course, we're using ascorbate all the time.
yeah, but DMSA passes the blood-brain barrier. And so, to me, that's a double-edged sword, because if you have a mercury deposit, say, in your kidneys or someplace else, the possibility of pulling it out is there, but the possibility of pulling it into the brain again is there. It's much like
But I've seen some remarkable recovery on Amy Holmes' protocol.
Yeah, this is basically my conclusion from this article, is that this has gone from a research problem to a clinical problem. That the issues of, how to treat autism or how to treat Alzheimer's disease are now just determining what clinical protocols work and which ones don't work. And it's just a matter of practice, trial and error, working out the details.
And in Parkinson's disease, there's a few doctors that have gotten famous, I forget the name, giving alpha lipoic acid as a intravenously and making significant progress. Does anybody know the name of this?
Oh, they give glutathione intravenously.
Right.
Perlmutters do.
Perlmutter is what I'm talking about.
It's
Yeah.
glutathione,
Glutathione, not
lipoic acid.
It's glutathione. That's right.
Yeah, but it doesn't work that well in everybody. There are some people that don't seem to respond to it at all, and other people that seem to respond dramatically. So it may be that some people, their ability to make glutathione is compromised. And it may be that other people have very high concentrations in their brain and not in their body, or vice versa. Until we have a way of monitoring mercury in the brain and being able to note how it's moving, we're basically judging this based on the end result, the clinical improvement that we see in people. Okay. So microtubules are long, rigid structures, and so they serve this kind of scaffolding, or not scaffolding, more girders and beams kind of function within the nerve cell. But there's a much more important function that they have that has to do with the transport of materials. And there are proteins, motor proteins, that the body makes that will grab onto these microtubules and start trucking down the microtubule. And there's the microtubules are polarized. Each one of the units are polarized, and so these have a direction. They have a negative end and a positive end. And one of these proteins goes from the negative end to the positive end, and the other protein goes from the positive end to the negative end. So these things serve as a highway system for the movement of material in the nerve cell. Okay? So these are like roads, and these motor proteins are like trucks, and they grab onto these sacks of material and drag them down these long pathways from one end of the neuron down to the end, or from the end of the neuron back to the center of the cell. And this is absolutely critical for the health and function, metabolic function in nerve cells. Okay? Now, if you take a nerve cell that's 1 inch long, which is, common, it's like 5,000 miles from this side to that side over that 1 inch, if you're going to make an analogy to this to the size of a road. But there may be raw materials that need to get there. So I don't know if anybody knows all of what kinds of materials are found in vacuoles and other types of components that could be carried. But one of the things that the motor proteins carry are microtubules themselves. So there may be microtubules that may be made in chunks like girders that may be, particular size, and they fasten these onto a motor protein, attach it to the microtubule, and send it down the line to the end of the cell where it's going to be used as a chunk. And as you remember, I said these things are polarized. They're negative on one end and positive on the other, so they're directional. Well, when the nerve cell first starts growing, it starts out always with the positive and negative ends oriented the same way. So that it's always growing outward. But at some point in the cell's development, some message comes back to the cell that says, okay, we've hit our target. And then a microtubule that's inverted goes down the tube. And so what I think happens is that you have motor proteins. So if you have a tube that's running the opposite way, it's like a U-turn. So the material is coming down, you have this gradient of material, and when you have this reverse tubule, you have everything coming back, and so you get a high point, and it creates a node in the nerve cell, which then you form a lot of different branches. And so that's how microtubules in opposite directions can be used to convey some information that causes nerves to grow in a particular way that wouldn't be if you only had it go one way.
Is this growth a function of what you're learning, or is it genetic?
Well, I can't say it's entirely genetic. It's what I would say is it's developmental. In other words, the genes control the program that starts it all out, but it's an active, interactive program. So how long a neuron grows depends on what it runs into. And so the program says, start a microtubule and let it grow in this direction. And if it keeps growing and keeps growing, you keep feeding the pathway, and as soon as a particular signal you're looking for comes back down the tube, then you change the program. It goes to step 2, and step 2 is you send a microtubule down the tubeway that's inverted. That goes down the tube. And then it goes to phase 2, and different things happen in phase 2. And then you have dendrite growth. So first you have axon growth, and then you have dendrite growth, and you have other smaller branching, and so the program is running, but how it actually connects up depends so much on what other cells are doing And that may be influenced by hormones, whether you have testosterone or not testosterone, or whether you have adequate levels of selenium or not, or whether you have you know, some kind of genetic abnormality. It could be a developmental abnormality based on alcohol exposure. All that's going to alter. It's an active program. It's like a symphony where everything that happens, you know, leads to the next step in the symphony. And everybody's end up being different. It's not just because the instructions started out different, but if you were to take yourself and clone yourself 100 times, you're going to find 100 totally unique people. They might look like you, but they'll have different personalities, different mental abilities, different IQs, different gifts, because the developmental part is unique. It's not, it's beyond genetics. Remember I talked about the heart disease? This is a sidebar out of my newsletter. That's the article that you have with you. And this is idiopathic dilated cardiomyopathy as connection to mercury. This is the kind of heart disease where, you know, teenage athletes drop dead in a basketball game, running from one end of the court to the other. And when they do autopsies on these kids, they find that their levels of mercury, this is, I've oriented this data according to its highest level here, where you have 6,000 or 22,000 times the normal level of mercury in these heart biopsies. And there are people with this kind of mercury that are still alive. They have heart disease, but they're still alive. Something is causing that mercury to accumulate. This probably will be found to relate to Alzheimer's disease in some way. We can figure it out. And mercury is at the top of the list. There's, you know, 22,000 times the level of mercury, 12,000 times the level of antimony, 250 times the level of arsenic. And then you're starting to get down to, you know, levels that are, you know, lower. But you look at aluminum, you look at, you know, silver, zinc, chromium, you find that they're all higher. So there's something about that everything is being concentrated, but mercury is being concentrated more than anything else. I also have this in a blow up to make it easier to see the numbers. It would be a little awkward to see that distance.
When they did that study, did they happen to notice if it was the first child in the family? I'm just thinking as the infant is a toxic dump type.
Yeah, but I don't know that there's a lot of correspondence with that. I know that the maternal environment is where the mercury all comes from, but kids that are born all have, I mean, there's not a huge variation in mercury over the course of a human lifespan. If you look at mercury levels, you find it's relatively constant. It's not like you can start out really, really low and go up. Fetal mercury exposure is definitely a big problem. Yeah. And I wouldn't be surprised if they study this, that they're going to find that mercury exposure is very, very strongly connected to Alzheimer's disease. But it would take a big study to see the statistical significance that's involved. And a lot of the studies have been done a bit small and done by dental organizations and published in dental journals and funded by and performed by people who you can raise questions about their motives and what they're really trying to accomplish in doing these studies. But if you do a big study and you do it without bias, I think we will find that amalgams are going to be found to correlate with Alzheimer's disease. I also wouldn't be surprised if the fetal mercury correlates with intelligence, because again, the intelligence of the human brain as opposed to other animals has to do with its branching. In fact, our neurons are much more branched than other animals. And that's why the human brain is especially susceptible to mercury, much more than your dog or your cat or a rat or a snail. Because we're so much dependent on microtubules.
In this table, could you clarify what IDCM means relative to the others?
That's the idiopathic dilated cardiomyopathy. These are the people here with that specific kind of heart disease, and these people have other kinds of heart disease
These people have a non-heart, they have a heart disease that's non-related to the oxidative stress, and these people have no particular connection to heart disease at all. So these are just comparing between different patient populations.
What does idiopathic mean?
It means they don't know what causes it.
Oh, okay.
It's just of unknown origin. And dilated cardiomyopathy means that the heart is affected, and dilated means that it's, swelling or expanding or something. Okay. Okay. So, you can see these levels here are just phenomenal, very high. 178,000 nanograms per gram. Yeah. But you can see, this different kinds of heart disease. These numbers are relatively low. An average person is probably going to be less than 10. And you start to get up in ischemic heart disease, 23 and 30, which, those are high numbers, but 178,000, it's just way over the top.
Steve, you mentioned one of your points is that not only do you need the mercury to get the Alzheimer's, but you have to have a bad immune system or be toxic. Can you tell from that chart, what constitutes bad enough immune system to be?
No, that's, in this particular situation, what's going on in IDCM, this cardiomyopathy, is, I suspect that the researchers who think that it's related to infection are right. That you've got some kind of bacterial or fungal infection, or probably viral, which is causing stress to the cell, which is, in a sense, sabotaging the cell's ability to handle mercury, and it's being deposited there, and it just keeps accumulating because of the infection. And maybe the infection sabotages the immune system, so this may actually be an adaptive mechanism of the microorganism to prevent the immune system from killing it, is by pulling mercury deliberately and concentrating it, and the immune system can't function with mercury because it's based on sulfhydrochemistry.
What's a shielding mechanism?
Could be, yeah. Yes, who's that, Bill?
I handed out an article tonight that related to mercury in landfills, and the report was that mercury deposited in landfills became methylated by the bacteria at work in the landfills. Now, if bacteria can methylate mercury, and if methylated mercury, as we know, is the most dangerous kind, maybe that's why, maybe methylated mercury tends to, you might say, pile up, accumulate more readily than other kinds, and if there is an infection of some kind in the body that could methylate the mercury that's there, that would explain it.
It actually would explain it in a negative way, because methylmercury is very mobile, it's lipid soluble, and it moves by diffusion quite readily, and it doesn't bind to sulfhydryl compounds very well at all. So it's more like a gas or a low molecular weight substance, and it moves. And so the reason methylmercury is dangerous is not because it poisons our systems, it's because it goes right across the blood-brain barrier. So you can get a lot of mercury in your brain quite easily, and then your brain takes the methylmercury and oxidizes it into ionized mercury, and then that starts poisoning all your systems. So that's the particular danger. Yes?
Yeah, a couple of other factors. The body's got to be made out of something, and when you are eating lots of sugar and processed foods that isn't loaded with zinc, manganese, chromium, selenium, and so forth, given an equal amount of exposure to heavy metals, that person who eats junk food is going to absorb and hold on to more heavy metals than the person with good nutrition.
That's true.
That's one thing. And you're talking about the glutathione thing, is that anything like viruses, bacteria, fungus, parasite, stress, all these things that produce free radicals and damage mitochondria make it hard for the mitochondria to produce enough NADPH and so forth to refuel the glutathione and so forth. And glutathione, I think, is more unstable in the oxidized form, and so all of those things could cause a breakdown of glutathione.
Oxidized glutathione doesn't bind to mercury.
Mm-hmm.
So it's the reduced glutathione that protects against mercury.
That's right.
Oxidized glutathione doesn't. And so if you lose control of your antioxidant, your reducing power in the body, then mercury toxicity magnifies dramatically.
Yeah.
What was the relationship between junk food and mercury?
Let me go through that again. Okay, there's a lot of trace minerals in the body that are used as nutrients, and a lot of them are divalent ions. In other words, they have a charge of 2 when they're. I guess I can bring that slide. Okay, it's in the newsletter. There's a periodic table, and so you've got copper and iron and zinc and calcium and magnesium that are all divalent, 2 charges. And lead and mercury are 2 charges. So they compete with each other. So you have a competitive effect between mercury and magnesium, for example, that can be pretty dramatic. And so if you have mercury levels that are here and magnesium levels that are down here, your mercury is going to be very much more toxic than if your magnesium levels are up here. Okay, so if you're eating junk food, your magnesium levels will be down because that's processed out. And you've got zinc and you've got other nutritional minerals that all have this competitive effect. Calcium would compete with lead, for example. So you get that same kind of thing. The calcium-lead ratio can be, you know, a wide variation, and the people who really have the problem with lead, where it really causes them symptoms, their calcium tends to be low.
Can we draw the conclusion that supplements of, say, calcium or other supplements that raise your level of these in your body will fight these problems?
I would say you can't assume that, but that in a general sense it's a good strategy, it's a viable strategy. But if you have, for example, a disturbed calcium-magnesium ratio, you may not be able to mobilize the magnesium to fight the mercury if your calcium is compromised. So there's all kinds of subtle relationships that may prevent you from being able to do that, or that might cause you to overload a system even more. So if you have a calcium deficiency and you take extra magnesium, it may cause negative effects that are not worth the benefit that you would get from protecting against mercury.
The literature says selenium is antagonistic to mercury.
Right.
In what respect?
Mercury selenide is inert. So when mercury binds to selenium, it ceases to be a problem for anything. See, if you look at the periodic table, you have oxygen. If you look on your newsletter, you'll see oxygen, sulfur, selenium. Okay, so they're all in the same family. And they all, they're reciprocal and competing relationships between them. So mercury binds to sulfur, it also binds to selenium. But when mercury binds to selenium, it won't bind to sulfur because it prefers selenium. The bond between mercury and selenium is so strong that you'll find a lot of selenium in the earth that's bound to mercury because of that reason.
Can that now be excreted? Or is it going to be somewhere?
Just sits there. Debris, cellular debris, molecular debris. But if it's not poisoning you, it's certainly not taking up very much space.
Yeah.
So that's one of the things that I have not researched yet is the role that selenium supplementation might play in detoxifying mercury. Most of the people that I've talked to have suggested that that selenium supplements do not produce a significant clinical response in any reasonable amount of time. That if there is an effect, it's fairly low level and tends to be long term. But there are issues of selenium that might be involved in mercury too, because in this there may be competitive effects where selenium gets selectively damaged. I mean, look at selenium down here. It accumulates less than anything else. And so maybe the high levels of mercury diminish the bioavailability of selenium such that the cell can't defend itself. And that may be one of the reasons that the mercury is able to accumulate to very high levels is because the selenium is sabotaged.
What kind of clinical effect were they looking for when they wanted to see if selenium therapy was effective in reducing mercury? Were they looking at urine mercury, or what were they looking for?
Usually, in a clinical environment, it's how people feel. You know, if you've got somebody with chronic fatigue syndrome, and, you know, does their energy improve when you give them selenium? But, you know, I suppose they could look at all kinds of data to try to see something.
I wonder if this is related to that. Was it Cushing's disease or something, that selenium deficiency in China that get a cardiomyopathy?
It could be that it's related to mercury.
And also, in Norway, reindeer are sort of the national beat, and they grow up north where they go through the snow and eat lichen that is high in selenium. Well, they got the idea that, well, we'll bring them down south so that we don't have to ship them down south. Well, then they started getting cardiomyopathy, the herds in the south. And one of the local vets told them that they should be feeding them selenium, and that stopped that. But the author of this made the comment that the farmer and the public were having the same cardiomyopathy but wouldn't take selenium.
But it's often politically incorrect to use veterinary products on humans. And that went on in New Zealand.
But let's see.
One of the New Zealand islands is extremely low in selenium. And the people figured it out and started using the sheep supplements and started using them on themselves. And a lot of their problems went away. And the government didn't like it, but, you know, it's like they can't stop the sheep from having the product, so they couldn't use people.
If Dr. Cathcart is right, then maybe that's the whole solution to this cardiopathy of unknown origin. Maybe the origin is selenium deficiency.
Well, a number of autopsies have been performed on athletes that have died on the field. I would imagine these last several, if they performed autopsies on them, they'd find a dramatic low level of both magnesium and selenium.
And which one is actually the problem and how much clinical benefit, they'd have to try that out. If they had somebody who they knew had IDCM and they gave them selenium supplements, they'd be able to see if their heart's capacity for pumping is improved. It'd be, you know, given the fact that they're so severely compromised, it probably wouldn't be very difficult. This is a study that shows that blood levels of mercury correlate very well with Alzheimer's disease. So you have these people here are controls, relatively the same, psychiatric and non-psychiatric controls, which we would use as a guide, and then we have late onset, people who are slightly susceptible to Alzheimer's disease, people who are averagely susceptible, and people who are more than average susceptible. Early onset Alzheimer's disease, almost 3 times the level of controls. So from this, I would presume that getting rid of mercury is a viable strategy. It may not be the best strategy, but it's certainly viable.
That's pretty high, though, 100 micrograms per liter.
Yeah.
I mean, in normal people.
Yeah.
Now, how much weight do you then give to the immune system problem? You have to both improve the immune system and get rid of the mercury, right?
No, I mean, I'm not.
Or are you saying
immune function.
getting rid of the mercury will improve the rest of their immune system, or is mercury itself the toxin that you're referring to as the only?
I don't think I have a clue as what it is.
Mike, but it's not the immune system; it's the detoxification system. That's where the glutathione and selenium.
It's probably one subset of the antioxidant defense system. I mean, if you look at glutathione, it's a pivotal antioxidant in the body, a critical antioxidant. It's irreplaceable, absolutely essential. And that mercury is specifically antagonistic to that part of the link. And so, you know, whether or not other antioxidants may play a supporting role, or whether it's really focused on glutathione or not, I really don't know if anybody knows. But I would say that in 10 years they'll study this and they'll probably say that glutathione plays more of a role in Alzheimer's disease than mercury does.
You mean as
a risk factor.
Really?
You mean
You mean the
positive?
low level of glutathione?
I mean, if you look at people who have Alzheimer's disease, some people have mercury levels down here, and some people have mercury levels up here. That the variation, the issue of who gets Alzheimer's disease has more to do with low glutathione levels than it has to do with high mercury levels. That's what you're going to find. That's what I'm predicting that they will
So
find.
take glutathione.
No, not necessarily. Taking glutathione does not necessarily raise your glutathione levels. And it may transport mercury into your brain, and break down and get transported into your brain where it increases your risk. I'm not saying it does, I'm just saying that we don't know. It's never been studied thoroughly enough. The mercury issue, if you look at grant money coming from the federal government to researchers to study various things relating to Alzheimer's disease, mercury is at the bottom of the list. It took, people who are renegades and non-establishment people looking into these questions to come up with this research. It's an end run around the government. I mean,
Steve, I don't understand though, because look, if you say that glutathione deficiency is a better, it's more of a risk factor for Alzheimer's than
I'm not saying that. I'm saying I think it is.
Okay, you'd say you think it is. That's your prediction. Okay, fine. But now the thing is, we know you can treat glutathione deficiency by taking whey and things like that that increase your glutathione levels. So I don't see why you're saying it's not a good idea.
I'm not saying it's not a good idea. I never did say it was a good idea. I said it might be a bad idea. It's not necessarily a good idea.
So you're just saying this is one of the things we're ignorant of.
If you raise your glutathione level,
yeah,
and you raise your mercury level more, is that a good thing to do? Does that make sense to you? That's a question.
Yeah. Well, see,
okay.
It's not, you're not raising your mercury levels. You're just kind of.
How do you know you're not raising your mercury levels? If you're taking cysteine or N-acetylcysteine or whey protein, how do you know you're not increasing the level of mercury in your brain? What data do you have to document that?
Well, okay.
There isn't any.
Okay.
I mean, we don't have a way of monitoring mercury in the brain. We don't do biopsies, brain biopsies on people, sequential brain biopsies to follow whether this is going up or down.
Well, for one thing, if it's true that glutathione doesn't pass the blood-brain barrier, then you can kind of imagine that glutathione would work to lower active mercury in the body. And that would at least be good, because then it'd be that much less mercury to work its way into the brain.
But it doesn't necessarily do that. I mean, one of the major excretion pathways for ionized mercury is through bile. Your body takes glutathione, binds it to mercury, shoves it out into bile, it goes down your GI tract, the microorganisms get toxic reactions to the mercury, they methylate it, it gets reabsorbed into your body. Is that of any benefit for you? So you've got this circulation problem with mercury.
So what you're basically saying is, that the problem is that we're ignorant about what to do about this.
Right. And so if you have dysbiosis in your gut, how does that change your mercury detoxification protocol? If you have very, very high fiber, high roughage, high vegetable diet, does that change your protocol versus eating a lot of meat and protein and fat? So we don't have answers to these questions. I'm telling you, this is all up in the air. It's got to be resolved as a clinical issue. That's the problem at this point. And you can go in and to your physician and say, I want to do this, and I want to do this, and I want to do this. But I'll tell you that you want to pay very close attention to possible signs of mercury toxicity when you undergo these kinds of things. Because if they move mercury into the brain, this could be a very bad thing. And so you don't want to wait until you have Alzheimer's disease to go, oops, I made a mistake.
Could you give me an idea of roughly how much money you think spent by the government to do research in this whole area?
I don't know. I mean, if I was interested in knowing that, I'd probably figure it out. But, I just am so morally offended by the way the government wastes money in scientific research that I just can't really look at it without getting sick. So I don't look at it. Yes.
Another strategy. There's a book by Kasdorf and Walker, Heavy Metal Toxicity. And Kasdorf describes how he has really thrown into remission advanced Alzheimer's cases with EDTA chelation. And it's quite a remarkable book.
What is the name of it again?
It's Heavy Metal Toxicity by Kasdorf and Walker.
Yeah, and there are other researchers who say that EDTA chelation therapy is risky for mercury because it does specifically transport mercury across the blood-brain barrier. That's what they say. So I can't say that there's much of a consensus. I think if you bring 10 chelation doctors into a room, then you probably have 11 opinions about mercury.
Now, you've mentioned that mercury is a possible cause. How about other heavy metals being the cause for Alzheimer's as well?
It's never been demonstrated. As a matter of fact, all the findings are negative.
Because based on what he just said, taking the EDTA, getting rid of heavy metals, improving Alzheimer's, seems there's some kind of correlation there.
Right. But lead doesn't poison the systems that are dysfunctional in Alzheimer's disease. The enzymes and the microtubules, lead doesn't affect them. It's mercury that does. Now, it may be true that you can reduce mercury's toxicity by removing cadmium, for example. So you have zinc, cadmium, and mercury on the periodic table right above each other, and cadmium does potentiate the toxicity of mercury. Okay. This is a study that was done by Dr. Haley, where he looked at the inhibition of beta-tubulin. So beta-tubulin is binding this GTP, and it's looking at how much of it, how much can it bind. And so if you have 100% binding, that means you have functional microtubules, and you have 0% binding, that means you have completely dysfunctional microtubules. Okay, so here's the concentration. If we look at mercury, which is the green bar, at this low level of mercury, you have very little inhibition. It's almost 100%. You go up here to 1.25, it drops 25%. So you get only 3/4 of your tubulin is functional. And the curve, it drops, fairly dramatically, and when you get up here to 5, it's way down, and it's fairly dramatic effect. If you look at zinc chloride added to it, you see an inhibition. But it doesn't happen to a significant degree back where mercury starts its effect. But when you get out here to 10 and 20, when the inhibition is maxed out on mercury, it's starting to have an effect from zinc. And cadmium's in between. So cadmium increases mercury toxicity. So here we have, this is mercury. The white circles are mercury by itself. The gray circles are mercury with a little bit of zinc added to it. And the black circles are mercury toxicity with a lot of zinc added to it. So this 10 micromolar zinc is here. So you're getting this much inhibition from the zinc alone, but when you combine that with mercury, it drops it from just a few percent all the way down to 50%. So it dramatically increases mercury toxicity. Now, I put a question mark here because this is not an in vivo finding. This is a test tube study. And if you are in a biological environment, you're certainly not going to have any zinc chloride around. Your zinc is going to be attached to other kinds of chelating agents and binding agents like cysteine or glutathione or who knows what. And so part of the toxicity of mercury chloride in these studies may be the fact that it's being added in a mercury form. And they may find that when they look at glutathione mercury compounds or cysteine mercury compounds, that it won't be quite this potent in its toxicity. It may come down out here a little bit.
Steve, question. The island of Guam has a very high Alzheimer, I understand, patient, very high incidence, but they feel a great deal of aluminum in the water. Now, you haven't mentioned aluminum. Are you familiar with that? Or?
Aluminum does not produce the enzymatic or the visual, the neurofibrillary tangles, doesn't produce any signs of Alzheimer's disease in animals or in cell cultures or in test tubes, in homogeneous, brain homogeneous.
Thank you.
There was some kind of stuff years ago about aluminum found in the brains of Alzheimer's patients?
Sure. Yeah.
So what about these people on renal dialysis where there was aluminum in the water they had a high incidence?
It looks like they didn't do aluminum in this study. But you do find it there, it's just not as much. The aluminum level is not as toxic as the mercury. So I mean, this study might suggest that you should be careful about zinc in Alzheimer's patients. But I don't think it's conclusive. I wouldn't be surprised if zinc might have an antagonistic effect against mercury, that there may be a competitive effect between mercury and zinc, and that by adding extra zinc, you make your zinc functional when the mercury would be sabotaging it. It could work that way.
What about activated charcoal, Steve? Have you heard of that, at least through the gut, if that will absorb heavy metals?
Yeah, it does, but again, whether or not it can be interfered with by microorganisms that will then take that adsorbed mercury and methylate it and make it reabsorb again, I don't really know the answer to that question. So it may be that it helps, it may be that it doesn't, it may depend upon gut-related conditions. This is an area that needs a lot of work. I mean, if the government were to take all that money it wasted and put it into studying this, we'd have answers pretty quickly if it was done by honest scientists. I mean, it's pretty simple. Gut-related issues, the liver, absorption, excretion.
The mercury in the gut is connected to sulfur.
Yeah.
Okay.
Or methyl group. See, the microorganisms want the sulfur, so they tie up the mercury, get rid of it on a methyl group, and then they can use the sulfur for their own purpose.
Okay.
Yeah. Okay. This is a layout of the genetic risk factors. How many people have heard that Alzheimer's is risks are genetically related? Okay. This is something that I discussed in the Seri newsletter, I don't know, back in 1994 or something like that. And there's a apolipoprotein, or apoprotein E, that comes in a variety of different forms, and these forms have a direct bearing on when you get Alzheimer's disease, what your risks are. So if you look at this grid over here, you see that people in this quadrant here get Alzheimer's before they're 70 years of age. People in this one get it between 80 and 90, and people up here get it older than 90, and people up here, nobody knows because they're so rare, they may not ever get Alzheimer's disease. You don't have enough statistical basis to judge that. And the actual factor that ties this ApoE protein to mercury is that the difference between ApoE2 and ApoE3 and ApoE4 is that ApoE2 has 2 cysteines, ApoE3 has 1 cysteine, and ApoE4 doesn't have any cysteines on 2 key positions that are different between these variants. So ApoE2 has 2 cysteines, ApoE3 has a cysteine and an arginine, and ApoE4 has 2 arginines. And arginine does not bind mercury, cysteine does bind mercury. So this is a way that the brain can excrete mercury. By producing ApoE in the brain cells, which then bind to mercury and get excreted and come down through the cerebral spinal fluid, that's a way that the brain can offload mercury. And by just increasing that slight advantage, that one can make the difference between having Alzheimer's before age 70 or after age 90.
Did you say that L-arginine is the precursor or the material needed to make the ApoE2?
No.
I'm sorry, I missed that.
The protein is actually made from all kinds of amino acids, but the difference between ApoE2 and E3 and E4 is that on positions number 112 and number 158 on this amino acid chain, they're different. So you find that there are 2 amino acids that are substituted that make these variants different from each other. And the substitution is that ApoE2 has cysteines in both positions, whereas ApoE3, which is the most common form that most of us have, is one of them is a cysteine and one of them is an arginine. And ApoE4, which is the one that's exposed to maximum risk. Alzheimer's at 70, if you have both genes, less than 70, if you have just one of those genes, it's between 70 and 80. Okay, so those people that have this type 4 are at the highest risk, and they have 2 arginines in those 2 positions. So the binding power of cysteine to bind to mercury is missing in those people.
So is that implying a detox?
Detox mechanism that's specific for the brain, a detox mechanism that's used by the brain. Yeah.
And this is ApoE is a gene allele?
ApoE is I mean, it's specified by a gene, but it's actually a blood protein that serves a variety of different functions. And what it does in the brain is not really known. But it has been measured there. The brain produces a lot of ApoE. And it doesn't serve the same lipid handling properties as the protein does in the blood. And the body does that a lot. Multiple uses for things. You have one gene that makes something, and you can use it for 5 different purposes. And so it turns out that there's probably some kind of basic housekeeping function that ApoE serves in the brain, and whether or not its primary function is for detoxifying mercury or it's a fortuitous accident, I can't say I know. Okay?
So where does the ApoE go? You're saying it detoxes, so how
It carries mercury down the spinal cord and out into the body. So it gets it out of the brain.
So it goes across the blood-brain barrier?
No, it flows down the spinal cord. So it's not going through the blood-brain barrier. I mean, or it's...
So it just goes down the spinal cord, with mercury in the spinal cord.
That's right. So I have a question that's come up, and what role does mercury toxicity play in spinal cord injury? A lot of fruitful stuff that can come down from this, from looking at mercury in this way.
Steve, does this argue for taking cysteine to detoxify?
No, this doesn't have anything to do with cysteine as a substance. This is cysteine within a protein. So it's bound in a globular mass, and there's a cysteine in there that it's not free.
But it's free to react with mercury.
Right. But taking cysteine is not going to increase your ApoE levels. Probably not. I would bet that ApoE is regulated by, independently of cysteine availability. Yes, I would predict that wouldn't be particularly valuable. But so the genetic model of Alzheimer's disease does relate to mercury, and this is, I think, one of the things that did raise my eyebrows and get me to look at this really closely was because of that, the fact that all of these different isolated findings with Alzheimer's disease can be tied together. Now, okay. One of the enzyme systems that's severely damaged in Alzheimer's disease is creatine kinase. And creatine kinase is highly expressed in the brain. And it regulates the backup supply of ATP. Turns out the brain doesn't use energy very smoothly. It goes through these, valleys and troughs of energy use. It's like power surges, and then it calms down, and there's a power surge, and then it calms down. And so this particular enzyme is extremely important because when ATP is available, it converts it into creatine phosphate, which is like a backup form of ATP, and then when ATP becomes depleted, it goes the opposite direction. It converts the creatine phosphate back into ATP here, where it can then do its work. It turns out this ATP can be converted into GTP, which regulates microtubules, which I've already mentioned. But ATP also is heavily involved in phosphorylation reactions. It turns out that a lot of phosphorylation abnormalities show up in Alzheimer's disease. As a matter of fact, the neurofibrillary tangles are made from tau protein, which is overphosphorylated. So the process that puts phosphate groups onto proteins and takes them off is severely disturbed in Alzheimer's disease, and this is one of the reasons why. A whole series of enzymes are modified by this system. And this is my analogy to explain how this works. So your body is producing ATP on a regular basis, which means you have a spray up here, and the water is flowing down the mountain, and your body's using that water like you would grow vegetables or something like that. And so this stream is the need for water is very high and then gets low, and very high and gets low, like it would be if you were growing vegetables in accordance with the season. And so how do you smooth out the need for water? You divert some of it when it's flowing into a tank, so that when the water stops flowing, it flows out of the tank and keeps the water supply going. Okay? So if the spring is not stable, you set it up this system and it smooths it out. And that's how the brain operates. Now, when you remember that video, when you were looking at the neuron, it seemed to kind of pulse. Notice that? That's because the phosphorylation and dephosphorylation systems are alternating back and forth. It's a 1-minute cycle, approximately 1 minute. The brain goes from being in a state of high phosphorylation to a state of low phosphorylation and back again. And so the cytoskeleton, the microtubules and the microfibers and other structural components, it's like they're tensing up and then they're relaxing. Tensing up and then relaxing. And this is how the neuron moves. It's how it grows, how it oscillates. It turns out that human nerves have this 1-minute cycle. And this is an important part of neural function. This is probably why the human brain consumes 20% of the body's oxygen and energy, and yet only occupies a very small amount of our total tissue. It is very high energy intense tissue. It's probably because our brains are critically dependent upon phosphorylation. It's not just for getting work done, but it's involved in information processing and structural stability. So a huge amount of energy is being wasted for those purposes. So it's like in Star Trek, they have their structural integration fields, and that strengthen the ship against shocks, and that's a consumption of energy to make the ship more rigid or whatever. And this kind of thing is going on in the human brain. ATP is stabilized by magnesium, and mercury is agonistic to magnesium. So that's another dynamic.
What do you mean by stabilized?
It's the ATP molecule is bound to a magnesium ion. It's like they travel together. The ATP is very, very negatively charged, and the magnesium is positively charged, and so it kind of wraps around it and serves to make the structure more stable.
To preserve
it. And magnesium is used as in a lot of enzymes for that reason, because it's needed for the ATP to work the way it does.
Produces the creatine. I've heard of it as a supplement.
Well, it's available as a supplement. I mean, the body makes it. It's probably on a feedback loop, where if the level drops below, the body starts diverting arginine. It's made from arginine. If you look at the structure here, the blue molecule is arginine and the black molecule is creatine, and this nitrogen, carbon, nitrogen ring, triangle system here is the same on arginine as it is on creatine. So it takes glycine and snips this end off the arginine, sticks it onto glycine, and that makes the creatine, and the SAM donates this methyl group right here, that black line. But the whole point is that creatine serves a very, very special function, because this phosphate here is attached to a nitrogen, which keeps it very energetic. And in ATP, it's attached to an oxygen. So this doesn't quite have the full amount of energy, but it has most of it. So it serves as a reserve, an energy reserve supply for the brain. And the article that you have details a whole series of different enzymes, like glutamine synthetase, which is inhibited. And what these enzymes all have in common is that they're sulfhydryl enzymes. So not only is glutathione a sulfhydryl antioxidant, but these enzymes are sulfhydryl enzymes. That they have a sulfur and hydrogen atom at their active site where they function. And here is a picture of that of an enzyme. Here's a sulfhydryl enzyme, and here's a pocket, a binding pocket, where one of its compounds comes in and sticks to it, and here, sticking out of this pocket, is a sulfur. That's the sulfhydryl compound of the enzyme. It's a critical part of its function. And so if something comes along like mercury and binds to that, it no longer works. This is what happens with beta tubulin. And why microtubules won't form with mercury is because this site here cannot be used to link up two tubulin molecules together to make the dimer, and then to link that to other dimers to make the the protofilaments, which then bind into the tubule. So this sulfur compound is critically involved in the infrastructure of the brain, and mercury attacks that specific weak link. It prevents the assembly of these parts into the brain structure. This particular compound, gliotoxin, has the same kind of toxicity that mercury does, but it's not in any way directly related to mercury. This is produced from fungus. And so candida infections, for example, can put people at risk for gliotoxin production, which will poison the same enzyme systems that mercury does. So you have this level of mercury toxicity and you have this level of gliotoxin toxicity that can produce destabilization of brain structures. And this is the molecule that can pass it around. The thing to understand about it is it has this sulfur-sulfur bridge, which is looked at from the side. You can see this thing poking out. Well, under reduced conditions, that bond is broken, and so you can have this can then fold down like that, and this part of the sulfur can stick into the enzyme site and bind onto the sulfur. So now you have this molecule here stuck onto the sulfur. It's like a barrier from anything ever getting in there. So it irreversibly binds that site, so it will never function again. This can be produced by fungus. It's something that could produce neurological symptoms. There are a lot of other compounds that can aggravate mercury toxicity as well. It's not very well understood. Dr. Haley in his laboratory does tests of teeth, and he finds that some of these teeth have incredibly high toxicity in these brain systems. What he does is he takes nerve cultures, and he makes homogenates out of them, and then he adds something to the cell and looks at what happens to the beta tubulin. Does the tubulin bind, or is it inhibited? And he uses that as an assay to say what's toxic to the brain and what's not toxic to the brain. Well, it turns out that there are certain kinds of bacterial infections that can produce powerful inhibition of brain tubulin. It doesn't seem to be that much related to mercury. So what's coming out of these teeth is it's very toxic, and it's not necessarily strictly a mercury toxicity issue.
Steve, a question. Is this analogous to collagen?
Collagen? No. Collagen does not form sulfhydryl based links at all. Yeah.
Steve, I'm wondering if you were to force or reduce the redox potential on that bond with massive doses of ascorbate, whether or not you might break them up.
I think you might be able to, but I don't know how you'd get that kind of ascorbate into the brain.
Intravenously.
Yeah, but you've got to go through the blood-brain barrier and then into the cells. You're going to have it's going to be dismuted by the diffusion, and I don't know that you'd get enough of it in there.
Well, you know, the cerebral spinal fluid apparently has 10 times the serum level of vitamin C, and if you use a micropipette on an axone or something in the brain,
it's another 10 times.
You have another 10 times. Yeah. So the brain is pumping it in there.
Right, but that's why I think that if you flood the body with it, you're not going to increase it as much because it's already flooded. The brain is deliberately trying to drive the concentration, and so if you add more, it's going to tend to be limited by the fact that you're not going to get. If you double the amount in the body, you're probably only going to increase the amount in the in the cerebrospinal fluid by 10 or 20%, and then in the brain cell itself by 2 or 5% or something like that, you're going to see a step down effect each barrier to the flow of its diffusion.
Steve, would you explain, in a sense, to the blood-brain barrier? We understand it's a barrier, but it just...
It's just a layer of cells.
A layer?
It's like skin that surrounds the brain and separates it from the rest of the body.
And what's the function of that? To prevent toxification of the brain?
Yeah. There are certain things in the brain that are processes that would be destabilized by things in the world. For example, alkaloids. Things that brain neurotransmitters are called monoamines, like serotonin or dopamine. And these things, there are a lot of amines in their food that if it crossed the blood-brain barrier would cause our neurotransmitters to get totally whacked out. So what happens is there's a layer of cells that says you can pass or you can't pass, such that all the amines in the brain are made by the brain where it can regulate its own supply, and anything that's coming in from the outside stays in the rest of the body. The brain is a very protected environment. And it's not just because of amines, but because of the microtubules. You have all this energy use, extremely high energy use. The reason that we're able to think like we are is because our brains are able to consume massive amounts of energy to do things other than just simple biochemical synthetic
And the
processes.
capillaries running through this blood-brain barrier that become clogged create strokes. Is that correct? Because of the lack of oxygen? Am I oversimplifying it?
Well, the blood-brain barrier surrounds those capillaries. So the blood supply that flows through the brain is on the outside of the blood-brain barrier. So the oxygen has to go through the blood-brain barrier to get to the rest of the brain. Does that make sense? Okay. I wanted to get back to dentistry here because this is not just an issue of mercury. I mean, we mentioned that gliotoxin can attack the same mercury-sensitive systems, and it turns out that anaerobically infected teeth produce toxins that also attack these same systems. And I can't say that anybody knows what these compounds are, but one of the things that Dr. Haley has suggested in his talks is that instead of getting methylmercury, you get methylthiomercury, because these teeth can break down methionine and cysteine, methionine in blue and cysteine in black, to produce methane thiol and hydrogen sulfide, and those things can then make mercury into intermediate compounds that are very toxic and that can move through the body. So they're not, held in a detoxified form. They become more virulent and can spread and poison enzyme systems. So he done some preliminary research on methylthiomercury and found that it's more toxic than methylmercury or thiomercury.
Where would this be occurring?
This would be occurring inside of your teeth or with a gingival infection. But mostly what he's found is that it's a risk factor for root canal teeth.
So you might be able to smell that, right?
Yeah. Yes. Hydrogen sulfide is a component of bad breath. Methane thiol is a component of bad breath. Aminoethanol is a precursor for acetylcholine. And CO2 is just one of the oxidized gases that can be excreted. So this is one of the things that he says that he's demonstrated that this does increase mercury toxicity, but he says this can't account for the root canal infected teeth. Something else is coming out in addition to this, which is very, very toxic. But he does, the good news is he has a testing system that you can use right now, or at least your dentist can access, where they can take a tooth that's been extracted and put it in water and then let it sit for 10 minutes or half an hour and take a drop of the water and add it to a brain homogenate and then measure how much beta tubulin is inhibited. Okay, so if you get 90% inhibition, it's a really toxic tooth. You get 5% inhibition, it's not very toxic at all. And they also have ways of collecting gingival fluid to test it for the same kind of toxicity, so you can know before you have your tooth extracted whether this is a risk factor or not. Okay, and the reason that you get anaerobic infections in root canal teeth is because the tooth has an internal structure of tubes that allow cells to get in to keep the tooth healthy and repair and rebuild it and allow the tooth to grow. And when you do a root canal, you chop out the core of the tooth and seal it up so you get all these tubes that get isolated. And there's no way for anything to get ALT Corporation, www.altcorp.com.
In fact, he's trying to get federal approval, FDA approval of some tests.
Yeah, these tests are not approved yet, and so they have to be done on an experimental basis. But you can talk your dentist into doing that.
I'd like to recommend a good book, Steve, that I read called The Great Root Canal Cover-Up. Have you read that, Tim?
Yeah, that's George Ming's book, and that's based on a lot of Weston Price's studies back in the 20s. So there's a lot of, there's controversy. He says take them all out, root canal teeth, but then there's a way that you can save a tooth by doing the biocalyx treatment. And Haley is doing a lot of research on that to determine.
You mean re-root canal? When you drill out the one that's there and redo it?
And then put a biocalyx in it. So he's doing a lot of research in that arena. So there's a lot of controversy and there's a lot of studies that are being done right now to determine the viability of the biocalyx versus the traditional root canal.
Yeah, but if you are in doubt and you have a health problem that may be associated with a root canal, you want to have some kind of way of assessing whether it's at high risk or not, and doing this gingival fluid test is a way of getting an idea. And if you get it tested and it turns out it's toxic, then you have the choice. Do you just pull the tooth and get rid of the problem right away, or do you try to rescue it and save it?
I have a question here. After I read that book, Root Canal Cover-Up, and I had to have 2 root canals for a partial because, as I mentioned before in this room, that my jaw started dissolving from too much pure ascorbic acid in there, the buffers. I believe that's the reason. I left the holes open. I don't have anything in there because I heard that if you put they put this gutta percia, which is some Philippine bark or something, or it's a kind of a gummy stuff, and then when it shrinks, it leaves an open cavity once you've capped off the tooth, and that open cavity is then allegedly the housing where the bacteria can grow, and then in turn will mask as a whole number of diseases, chronic fatigue, Epstein-Barr, arthritis, on and on and on. And so it has multiple personalities, whatever bacteria systems.
The gutta percha is a polyisoprene. It's rubber.
It's a rubber. It comes from a rubber plant in Indonesia. And the biocalyx system doesn't use the gutta percha. So there's
it's like kind of a cement or a concrete or something.
Yeah, they use so a lot of endodontists now are gone to MTA, which is mineral triglyceride aggregate, which is Portland cement. And they found out that the body around the root canal, the bone, the cementum, ligaments around the root canal